Students, we had previously told about the Ames test and how to detect the carcinogenic as well as the mutagenic property of a substance, and we told the relationship between the carcinogenicity and the mutagenicity of a substance. And coming to the Ames test, sometimes we can see that we use rat extracts for the Ames test. In the previous class, we had mentioned that Salmonella typhimurium strains are being treated with the mutagen, and they are being plated on two different medias, one which contains the mutagen and one in which the mutagen is not being present, and you check the colony, the extent to which auxotropy is being reverted under the influence of the chemical agent. Now, but some chemicals tested might not be mutagenic unless they are being transformed into another an active form. So, in animals, such transformations occur in the liver, and indeed many known carcinogens. Now, in the case of aflatoxins, they are usually not carcinogenic until they are modified by the liver enzymes, and that function to destroy toxins and other materials they might be then further circulating in the blood. However, in some cases, these enzymes would transform chemicals into more dangerous forms. For this reason, a mammalian liver extract is often added to the molten top agar prior to plating the bacterial cells used in the Ames test. And the extract converts the potential mutagen into the derivatives that readily react with the DNA, mimicking the enzymatic transformation that occur in mammals. And carrying out the Ames test with or without the addition of the extract shows which compounds have intrinsic mutagenicity and which need activation after uptake. Despite the use of liver extracts, only about half of the potential carcinogens are detected by the Ames test. So, if you are going to consider some chemicals, as we mentioned earlier, they have been found to be carcinogenic and mutagenic only after they enter the body and they undergo some transformation. Now, in the case of aflatoxin, usually they become carcinogenic unless and until they have been treated by the liver enzymes, which will destroy the toxins and other materials that have been circulating in the blood. So, in such cases, you need to see the action of, when you say that a chemical is not being found to be mutagenic, you should ensure that it is not mutagenic even in the presence of an enzyme. If you just say it is not mutagenic, it wouldn't be sufficient enough, because sometimes when that chemical goes inside a body, probably it would be converted into a mutagenic substance under the influence of the enzymes of our liver. So, to ensure that the chemical is not mutagenic even within the body, we do an Ames test in which probably along with the mutagen, we will also add some you will also add some liver extracts. Now, when you are doing it in rat, of course, you take the rat liver extracts in the Ames test. And if you sometimes you can also get any mammalian liver extract can also be used for this. Now, this is a picture which is showing that. Now, you can either take a rat liver extract, and Salmonella strain is being histidine auxotrop is there, and you put a possible mutagen into this and you plate it and you incubate it. And so, there is a mutagen in this, but here you take a plate and in that what happens, you put the non-mutated or the wild type Salmonella strain here is being incorporated. But after incubation, probably you can have some control trait revertants that which we had mentioned earlier, and here there will be high number of mutants, mainly because of the presence of this mutagen. So, here you are also incorporating the rat liver extract, mainly because if this mutagen has to be converted into a active mutagen, or it might not be mutagenic at the beginning, under the influence of the rat liver extract, probably it would be converted into mutagen. So, to assess whether it is getting converted into a mutagen by the action of the liver enzymes, we incorporate the rat liver extract also along with the Salmonella strain. Now, this incorporation can either be, this rat liver extract incorporation can be done either here, or even if you want, you can put it into this plate. There is a media here with this also being which can be incorporated with the rat liver extracts. Now, this is another version of the same experiment what we are talking here. You are taking a liver and you are taking a rat and you are injecting it for the animals with Aeroclone, and what happens, the enzymes of the liver are being taken, okay, to carry out and to carry out the metabolic process of detoxification, and then you get the sample here. The rat liver is homogenized. This is a blender which you see here. It is being blended in this tube and you get the centrifuge over here. After centrifugation, you get the centrifuge product over here. So, that liver enzyme along with the microbial strain and the X is the mutagen, and you mix it together and you put it into an agar plate which is being found to be what? Deficient in histidine. And here there are lots of organisms which are being formed, and same time you take the extract from the liver extract of the rat and you have another strain. This is a different strain of Salmonella typhimurium, and then you have the mutagen, and then you again do the plating here onto a minimal media which lacks histidine, and you have S9 here. You have strain 1 or 2 considered to be as control, but you do not take the mutagen and you check whether how much are the histidine natural reversions. So, compared to this, when you have any mutagens any revertants over here, you can, that is more amount of revertants are being present in the, what do you say, in the media of the plate of the organism, media containing the organism which was being cultured in the presence of mutagen. So, then it indicates that this chemical is being found to be have a mutagenic nature. I guess it is clear to you. The purpose of using a rat liver enzyme is to ensure whether the conversion of the chemical substance into a mutagen is possible only under the influence of a liver enzyme. That is, we do expect that if the substance was not a carcinogen or a mutagen initially, but once it enters into your body, later on is it converted into a mutagen under the influence of liver enzymes. So, to check that, probably you take liver enzyme extracts, okay, and that is also being treated along with the chemical which has to be analyzed. So, this was one method, or you can either incorporate it into this media also. I hope the Ames test, the conventional Ames test technique using Salmonella typhimurium is clear to you, and we will be continuing some modifications of the Ames test in the coming sessions. Thank you.